If you can see and hear me clearly, can you raise your right hand? Awesome. And I truly mean awesome. Through my words, I have encoded a program into you, an AND logic gate. It's a relatively simple program with 2 inputs and 1 output. The first input is the ability to see me. The second input is the ability to hear me. You need both the first input and the second input to get our output of hand raising. So we have an AND gate. It's because of our biology that we're able to interpret these acoustic signals and waves of light and execute this program encoded in a single sentence. You can imagine with more complex biological programs, we get more complex biological behaviors, like walking, jumping, or giving 5-minute talks about science. You can even imagine encoding new behaviors, like studying cancer cells in response to drugs to develop new therapeutics. If you think this is the coolest thing in the world, you just might be a synthetic biologist. Hello, I'm Emily, and I'm a synthetic biologist. So, what is synthetic biology? It's what happens when you let engineers into the biology department. It's the art of seeing biology as a composite of parts that can be manipulated to encode new programs with new functions, such as raising your hand in response to an auditory input or spying on cancer cells in response to a therapeutic input. So let's say we want to study cancer cells responding to a drug. How do we do this? One of the best ways to study cancer cells is by the proteins they produce, since proteins determine whether cancer cells spread, bind to drugs, or die. But studying cancer cells in their native environment is difficult because there's a lot of other cells also producing proteins. But if we were able to tag proteins, we could look at where they're coming from. Luckily, we have something that does this, which I call the protein spy. So now we have a system to create an output of protein tagging. However, if we tag all proteins, we're back to where we started. So we need to create a program with inputs so we can target those cells specifically that are responding to the drug. But proteins don't have brains, so we can't just tell them to do this. Instead, we have to encode it in a gene. Now, as synthetic biologists, we design our programs in similar ways, thanks to the central dogma of molecular biology, which states that genes are used to make mRNA, which is then used to make protein. With several biochemical pathways that interact to regulate these processes, there are a lot of means of control. But let's keep it simple. Let's imagine a switch with which you can turn the production of a protein on or off. Biologists might call this a promoter, and they can respond to many different types of signals, like the presence of a cell, a protein, or a molecule. With these switches, we can now encode programs into our proteins so that they can respond to an input and produce an output. But life is complicated, and sometimes we need more than one switch. In my thesis work, I designed protein spies split into 2 pieces, where each piece is controlled by its own switch with its own input. In this way, I need both inputs present for both pieces for my protein spy to work. So this is like the AND gate I mentioned earlier. I also designed a new type of switch, a folding switch, where only in the presence of a drug does my protein spy work and tag proteins. In this way, I create a 3-input AND gate, where I need the drug present in order to activate my protein spy. So here I show 2 protein samples. The ones on your right have the drug present, while the ones on your left do not. The dark bands indicate where the proteins have incorporated the tag, so you can see that in the presence of a drug, the proteins get tagged. So now, while my thesis work has been focused on developing tools to study biology, these same principles are being used to develop new therapies and personalized medicine. For example, the FDA has recently approved a new cancer therapy where designer immune cells are injected to cure cancer. Synthetic biologists are engineering those immune cells now so that they too have logic and compute multiple inputs, just like my protein spy. So by engineering logic into biology, we create smarter biological tools so that maybe one day curing cancer will be as easy as raising your hand. Thank you.